Hello everyone, my name is Marcus Walton and today I'll be presenting my Unit 2 project exploring protein structure and function of the hemoglobin beta mutation. Throughout this presentation I will explain how a single DNA change can dramatically impact protein structure and function while linking molecular biology to real-world health outcomes. The goal of this project is to show that protein structure predicts function and that even a single amino acid change can cause significant disease. My model compares healthy hemoglobin beta to the mutated form responsible for sickle cell anemia, illustrating this core idea. I studied hemoglobin beta, or HBB, focusing on the E6V mutation, in which glutamate is replaced by valine at position 6 of the beta chain. This single hemoglobin gene change on chromosome 11 causes sickle cell anemia. Although only one DNA base changes, this tiny substitution directly alters how the red blood cell protein folds and functions. The hemoglobin beta's normal function is to transport oxygen. It forms part of the hemoglobin tetramer, which binds oxygen in the lungs and releases it in the tissues. It also carries carbon dioxide back to the lungs. Hemoglobin's flexible, round shape keeps red blood cells smooth and deformable, allowing them to move easily through veins and deliver oxygen efficiently. The hemoglobin beta is in the red blood cell cytoplasm, made in bone marrow, present in oxygen-dependent life forms, and acts as a transport protein. The E6V mutation gives hemoglobin a new unwanted property. It tends to stick to other hemoglobin molecules. Normally, hemoglobin stays soluble and moves freely in water, but replacing a hydrophilic amino acid with a hydrophobic one makes the molecules clump, especially when oxygen levels drop. In normal hemoglobin, the DNA codon GAG produces the mRNA codon GAG, which stands for glutamate. The mutated gene changes to GTG, producing valine. This substitution shifts chemistry and begins the changes leading to sickle cell disease. In my model, the orange pipe cleaners represent correctly folded globin protein subunits. The blue pipe cleaners represent glutamate, hydrophilic residues keeping hemoglobin dissolved. The blue pipe cleaners represent glutamate, a hydrophilic residue that keeps hemoglobin dissolved. The circular red pipe cleaner represents a round, healthy red blood cell. Because hydrophilic residues interact with water, hemoglobin molecules remain separate and soluble, keeping the cell flexible and oxygen-rich. In the mutated model, orange pipe cleaners show globin, and yellow pipe cleaners highlight valine mutations. The rose sticky notes show the long fibers that form when hemoglobin molecules stick together. The red curved pipe cleaner represents the distorted, sickle-shaped cell. Valine's hydrophobic nature causes the molecules to clump, producing fibers that deform the cell and block blood flow. This example illustrates my key point that a single base substitution at the DNA level can dramatically change protein function and ultimately human health. Normal hemoglobin stays dissolved and flows smoothly throughout the bloodstream, while mutated hemoglobin sticks together and forms clumps. As a result, healthy round red blood cells move more easily throughout the veins, but sickle cells become stiff and can block blood flow, leading to pain and tissue damage. Glutamate is a polar, negatively charged, and hydrophilic amino acid that forms bonds with water, helping hemoglobin remain soluble. In contrast, valine is nonpolar and hydrophobic, causing it to stick to other hydrophobic molecules. This difference in polarity is a key factor that leads to sickle cell disease. Normal hemoglobin forms hydrogen and ionic bonds that help it stay dissolved in the bloodstream. In contrast, mutated hemoglobin develops hydrophobic interactions between valine residues, creating sticky patches that cause neighboring molecules to clump together. These long fibers make red blood cells stiff and distorted, leading to the characteristic sickle shape. Protein structure directly determines its function. In the E6V mutation, hydrophilic glutamate is replaced by hydrophobic valine, creating sticky regions on the surface of the hemoglobin molecule. At the protein level, hemoglobin molecules stick together and form rigid fibers. At the cell level, this causes the red blood cell to lose their flexibility and take on a sickle shape. At the organism level, the reduced oxygen transport results in chronic anemia, severe pain, and fatigue. This chain of effects demonstrates how a single microscopic change can lead to a serious health disorder. At the protein level, hemoglobin molecules stick together and form rigid fibers. At the cell level, this causes red blood cells to lose their flexibility and take on a sickle shape. At the organism level, the reduced oxygen transport results in chronic anemia, severe pain, and fatigue. This chain of effects demonstrates how a single microscopic change can lead to serious health disorders. Although the pipe cleaners and sticky notes cannot capture atomic level details, molecular movement, or the exact process of oxygen binding and release, my model effectively demonstrates how a single amino acid change can alter protein structure, distort red blood cell shape, and disrupt its biological function. The model I created is a simplified representation of a realistic hemoglobin.